Lastly, we have the multiple alleles. Mendel's experiment suggested that just two alleles exist for a gene, and these alleles appear in just two versions, a dominant or a capital letter, and a recessive or a small letter. Today, we know that many alleles may exist for the same gene in some genes. This now makes many possible dominance relationships in many organisms. An example of multiple alleles is coat color in rabbits. Here, 4 alleles exist for the C gene. We have this allele, a capital C, a small c with a superscript of CH, a small c with a superscript of H, and a small c. These alleles here are arranged in descending order of dominance, which means now that the capital C is the most dominant among the 4, while the C with a superscript of CH is recessive from this, capital C, and dominant over this 2, over the C with a superscript of H and the recessive C. And then, while the C with a superscript of H is recessive from these 2, but this one is dominant over the small letter c. And this small letter c is the recessive of them all, or the most recessive of them all. These different alleles may combine together. The wild-type version with a genotype of both, or homozygous for the allele capital C, is expressed as brown fur. The chinchilla phenotype with a genotype of capital CH, with a genotype of C with a superscript of CH and a C with a superscript of CH is expressed as a black-tipped white fur. The Himalayan phenotype with a genotype of C with a superscript of H and C with a superscript of H has a black fur on its extremities, while there is white fur elsewhere. Finally, this is the albino, or the colorless phenotype. It has a genotype of both the recessive Cs. So it is expressed as having white fur. Since there are dominant and recessive here, when a rabbit has an allele of this, a capital C, and another allele of a C with a superscript of CH, the phenotype would be brown, since C is dominant over CCH. So this C masks the expression of this allele CCH. These relationships may exist together in certain characters in organisms. For example, in humans, the concepts of multiple alleles and codominance are observed in our ABO blood types. So in humans, there are a lot of blood groups, but the most common is the ABO blood typing and the Rh positive or negative. For us, it is really essential to identify the first the blood type to be transfused to the recipient and check if it is compatible, because incompatible blood types result into an immune response in the recipient, which might cause even death. In ABO blood group, the blood is determined by the I gene, which has 3 possible alleles: the I with a superscript of capital A, the I with a superscript of capital B, and B small I. These are the 3 possible alleles, and it exceeds the idea of Mendel that there are only 2 possible factors or alleles. So this is where the concept of multiple alleles enters. So these are the 3 alleles. These 3 alleles here form different combinations of genotypes, which determine now the phenotype or the blood type of an organism, which I will be presenting in the next slides. So the allele I with a superscript of A encodes an enzyme that inserts antigen A on the surfaces of red blood cells. So when this allele combines with another I with a superscript of capital A allele, it results into a blood type of A. While the IB allele encodes an enzyme that inserts B antigen on the surfaces of red blood cells. So when this allele is combined with another allele of the same type, this results now into a blood type of B. These alleles, the I and the B, are dominant over the small I. So a person with a genotype of I and a small I, or homozygous recessive Is, it produces now neither antigen A nor antigen B on the surfaces of the red blood cells. So that person's blood type is blood type O. And a person with a genotype, with a genotype of IA and small I still results into a blood type of A. While a person with IB genotype and I results still into a blood type of B. Now, what if IA and IB have combined? So the genotype now of the person will be these two, IA and IB. What would be now the possible phenotype or the blood type of the person? The blood type of the person would become AB, since IA and IB are co-dominant with each other. So they are both expressed, or the A antigen and B antigen are A antigens and B antigens are both found on the surfaces of the red blood cells. So it results now into a phenotype or a blood type of AB. So to give you now the visuals, we have here these figures. Again, if the genotype of the organism is homozygous for A, which is this one, and this results into a phenotype or a blood type of A, since the expressed enzyme only inserts antigen A on the surfaces of red blood cells. So in this figure, these are the antigen A's. Yeah, these are the antigen A's. These are the only antigens inserted whenever the organism has this genotype. Also, if the genotype of the organism is IA with a recessive I, it will still result into a blood type of A, because the recessive I, which is this one, neither produces enzyme that inserts antigen A nor B on the surface of red blood cell. So it is being masked by the expression of the this allele. On the other hand, if the organism has a genotype of homozygous genotype of this one, IB, it results into a blood type of B. So since the expressed enzyme only inserts B antigen on the surfaces of red blood cells, so we can only see B antigens on the surfaces of the red blood cells. So in these figures, in this figure, in the second figure, these are the antigen B. So these are the only antigens inserted on the surfaces of of the red blood cells if you have this genotype. And then whenever this is paired up with the recessive I, this would still result into a phenotype of B, because this does not encode any enzyme that inserts antigen A nor B on the surfaces of the red blood cells. Therefore, this masks the expression of this allele. Okay. The third one is the genotype of IA and IB. This results into a blood type of AB, since the enzymes that insert A and B antigen on the surfaces of red blood cells are expressed. So both antigens A and B are found on the surfaces of the red blood cells. Again, this is an example of codominance, because both of the phenotypes, or both of the phenotypes appeared on the traits of the offspring. So in this case, whenever a person with a blood type A and a blood type B reproduce their offspring, most probably would get a blood type of A when they are AB whenever they are homozygous. So this would be the structure, or this would be the antigens that are found on the surface of the red blood cells of their offspring. And then lastly, if the genotype is homozygous recessive, just like this one, so what you can see there on the last part, this results into a blood type of O, since neither enzyme that insert A nor B antigen is produced. So there is no antigen A nor antigen B that is found on the surfaces of the red blood cells. So that's now the blood type O. So those are the three other dominance relationships that Mendel was not able to find out. Those are the incomplete dominance, codominance, and multiple alleles. These different relationships have added many possible variations in the traits of an offspring, which has increased genetic variation of organisms in specific species.